that is oxidative phosphorylation with our electron transport chain, and then comes our last exam. Okay? So while I'm uploading all of this, or it's actually already flipped on, I'm pulling it up, I'll go ahead and talk about the exam. Exam 4, you're welcome to come see it. I will be in my office hours after class. So I do have a lab meeting, right after class at 11:30, but I will be back in there probably about noon-ish, right? So definitely will be in my office from 12:30 to 2:30 because I have a lot of letters of recommendation that are due today that I will be working on for my students. So I will be in there. All right. I will tell you, I'm feeling a little raw today. One of my very, very close friends passed away, and so I'm not in a very good day. So if you're going to come and speak to me in my office, please do it with respect, okay? Got caught a couple of times yesterday, and I really just want y'all to be respectful when you're speaking to me, okay? Because it just, you should get to be nice, right? I realize some people were disappointed in how they did on exam 4. I'm very sorry for that, right? But that being said, I own no ownership in that. So how you do it on exam is you. It has nothing to do with me, doesn't reflect on me in any way, shape, or form. How you do on exam 5 also don't reflect on me. Okay? Why? Y'all, I went out of my way this semester to do things for you. Okay, I hired SI for you, which was something that we hadn't had in a long time, but it's very, very good. I gave you extra credit. I gave you extra points here and there on an exam, right? And then we're going to drop an exam. It's a whole bunch of other things, right? To try to help you, okay? So I'm always willing to answer questions on email and things like that, right? So you are always welcome to reach out to me or to the TAs. We do have a private tutor as well, okay? So you have all of the resources there, all right? Everything is free game on an exam, both on the lectures, slides themselves, and conceptually. You can never make everyone happy. Some people prefer straight memorization. Some people prefer conceptual questions just because they're good at understanding concepts but not really memorizing things straight off the slides, right? This past lecture, or past exam, almost predominantly like off the slides, right? That's just because I made it early, so I just copied and pasted in things, right? That being said, sometimes wording get funky or whatever, sure, right? So it was brought to my attention yesterday that the wording on kind of like question 40 was just a little awkward, and so I gave it all back to you. Okay, so you'll have 2.5 on there, added back. That being said, we did have some hundreds on the exam, so congratulations to those people, right? And we actually had people go up quite a bit, all right? Now, for that being said, there was some people that went down, right? This was not your best exam, okay? But there were some people that went up. The average stayed about the same. We stayed pretty much close to the same almost every time. We were a few points lower. If you look at the information by clicking the column, which is what I'm worried some of you are doing, many people elected not to take this exam, and so they're all in their zeros. So if you click it up and it shows you like the average, it's taking all of the zeros into account. Okay, so please don't freak out. The average is not that. That is just with everybody in there, and I put the zeros in there for people that chose not to take it. Okay? So I know that people were worried because they thought the average was super low. It really was not. Okay, I think it was maybe 2 points lower, but like I said, a lot of people did better. Some people, God love you all. Some people, I swear, I've had the same grade on every exam all the way through. I appreciate your consistency, right? So some people, they just always do the same thing. It don't matter what they're doing, but by hook or by crook, we'll get you there at the end, okay? If you come all the time, right, y'all, I will work with you, right? That's what I was just telling someone. I know who's here all the time. I know who works hard, okay? So just do your very best. Now, as far as exam 5, in order to benefit you, I have moved it to Thursday. Why? Because we need to get through the material and have time to cover it. Not only am I going to move it to Thursday, but I am doing the review. Me personally. So I'm going to take my time and do the review for you. Okay? And that will, A, give my undergrads a break because they have their own finals, and B, will let me see kind of how you're doing. Okay? Let's see, what exam 5. The reason why, let's get into exam 5 real fast, because that's a lot of things that are happening on the group me's, y'all. I hate group me, I think it's toxic, but whatever. Y'all feel like, get on your bitch all you want up here, okay? Do I hear it? Yes, I do. Okay? I don't care. All right? I figure people are just venting, right? That's fine. I leave here and I vent too, okay? So that being said, exam 5, when is it? It's Thursday. Normal time, y'all, class don't actually end until the 9th. Y'all realize that, right? The semester doesn't end until the 9th. So I have it within my authority to move it back to the 8th because we're still supposed to be in class. Okay? So please don't get pissed off that I moved it to Thursday. I talked to Dr. Shrikant yesterday and she was like, that's still a class day. I was like, yes, I know, right? So I'm sorry if you've done something to where you can't be here. We will try to work something out. But that being said, it is still in session, the normal session, right? We're not even into final exam week yet by that point. What else? Exam 5. What is it? It's cumulative. When I say cumulative, what does that mean? That means you just have to know all the stuff you knew before. What does that mean? It's just that we talk about side chains, coenzymes, things like that. Y'all, I don't ask you things from again from exam 1, right? Now, why is it not dropped? Okay? There's several reasons why it's not dropped, and actually we might have some new ones yesterday. One of which, you need this material. This material actually is going to be on the MCAT and several other things. You need the material, so it's always taught and it's always tested. The other reason is that there's extra things on the back that I want you to fill out for me. Okay? So that's always feedback that I always do every semester, and I want it. Why? Y'all were kind of a test case this semester where we kind of floated, right, based on how y'all were feeling. Some of you absolutely hated that. That's fine. Write that down. Don't say you hate me. Just say that you don't like that there was a dates on there. Okay? That's fine. Why? Because this was a test case. Why? In the fall, I don't do that. In the fall, it's predominantly STEM students. In the spring, it's off-cycle, what they consider off-cycle, and we have a lot of different backgrounds in here. Okay? So we pump the brakes at the very beginning so that everybody could kind of catch up, right? I realized that threw people off, they weren't liking it, whatever. Fine. Write it down, I'll change it, right? That's fine. We try something, scientists, we try things, they work or they don't. If it doesn't work, then that's okay, right? I don't take that personally. So, you just need to let me know, and I'm happy to hear it. All right? So that being said, exam 5, you will have to take it. Is it dropped? Historically, no. Why? Out of the thousands of students I have taught, and by the way, you are in the thousands now, out of the thousands of students I have taught biochemistry, there's only been a handful of people that this was ever their worst grade. Do you see what I'm saying? It's almost everybody's best grade. In the event that it is your super ass worst grade, I'm obviously not going to tank you if you had an A the whole way through the semester and then all of a sudden you did shitty on the last exam, right? I wouldn't do that, okay? So yes, in that case, then I would drop it. But it just normally has not been a thing. So historically, I drop it. I just say it. All right? Any questions about any of those things? I will tell you that if you don't like conceptual questions, exam 5 is going to suck. Why? Because I only have 2 things to really talk about on it, and so a lot of it will be conceptual. Some of it will even be experimental, but we'll be talking about it. Okay? All right. I feel like I've vented enough and I'm ready to go. Okay? Yes?
Question. So you said there's 2 things for exam 5?
Yeah.
Why is cumulative?
Yeah, like you still have to memorize, you still have to know all your amino acids and catalysis and what coenzymes are and all the other stuff, right?
But not like the whole material.
That's what I said. I'm not going to go back to like lecture 1 and start looking for stuff. Yeah, no. That's more work for me too, so I wouldn't do that either, right? All right. So let's get to the TCA cycle. Y'all always like to say Kelvin cycle, whatever, right? This is the Kelvin cycle or the TCA cycle, tricarboxylic acid cycle. All right. What is it? This is oxidative respiration. So we ended with glycolysis, and glycolysis we said was anaerobic because it actually precedes accelerates, right, the rise of oxygen in the atmosphere. Okay, so it's something ancient. And we said all of our cells do it. Pretty much every living organism has some kind of form, sugar breakdown. So let's talk about this. Y'all, talking about predicted light oscillation, I love it. It's plant-based. No one teaches it but me. That being said, sometimes I do give bonuses off of it, so if you want to go look through it, right? It's kind of an interesting thing, but it's talking about plant metabolism whenever you're looking at germinating seeds. Okay? So here is the pathway that we've been talking about. We got all the way to pyruvate, right? We said pyruvate has 3 different potential fates based on what? Oxygen availability and kind of your metabolic needs. If you have ATP rich, right, you have a high energy status in the cells, then what happens? We don't go through the TCA cycle, right? We said ATP kind of inhibits pretty much everything, right? Because you don't need to go through it if you're already energy rich. What would you do with that? You'd probably break it off and do some anabolic pathway, right? So let's look. We have pyruvate. We come in, we have acetyl-CoA. You're going to learn little small descriptives. I saw a lot of people wrote on the back of their Scantron their 10 steps for glycolysis. Believe it or not, that was almost a bonus. So, I know, sad, right? You probably knew it. So, I'm going to give you like 8 steps. We'll shorten them down for you to memorize these 2. Okay, I think that always helps, is that repetition in those 8 steps. So we're going to come in with acetyl-CoA. Now, acetyl-CoA is not really per se a step. It's kind of a preceding step, and we'll talk about how we get to acetyl-CoA from pyruvate. But we basically just say 2 carbons come in. Okay? And so that's kind of how we start out. Okay? Now that's not really reaction 1, but that's just kind of the precursor to it. Okay? So we say 2 carbons come in. Then we have citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA. Succinate's a cool one because succinate dehydrogenase is actually part of the electron transport chain as well. And so it's talking to both pathways. It's pretty cool, right? Then we have fumarate, malate, and oxaloacetate. So succinate, fumarate, and malate all just derive oxaloacetate. That's all they're for, is those reactions and those things. Those are all intermediates to go back to the very beginning. Now, that being said, that very beginning step, that oxaloacetate, is what's pulling the first reaction forward. That's why it's a cycle. Okay? So it's with, did I ask this question? No, I guess that was body. All right. For something to be considered cyclic, the product is actually also the first kind of reactant. Okay, so the product of the final path is also the initial thing. Okay, so oxaloacetate is part of that first step when we come in. All right, then we're going to talk about electron transport chain, and we'll go to oxidative phosphorylation, which is one of my favorite things because I think it looks pretty cool. So, I'm not going to read these are for y'all. Completion of our TCA cycle. Let's talk about oxidation of succinate to, did that pull up the wrong one? Yeah, it did. They look the same on the very beginning. That's just the same slide. So, let's go to there. There we go. Nope, 24. That's going to be lecture. There we go. Okay. Alright, here we go. Sorry. Lecture 34. Alright, so citric acid cycle. So we kind of already said this. We have aerobic metabolism, right? Why do we say we have aerobic metabolism? Because now we have oxygen available. In the beginning, right, it was mostly sulfur gases and things like that, and so things were anaerobic. Okay, so aerobic organisms use oxygen. What do? They use oxygen to extract energy from nutrients, and then oxygen is our terminal electron acceptor. You always hear that, right? Oxygen is the terminal electron acceptor. Respiration. Respiration is just that, oxidation, right? We're using oxygen. Now, we said fermentation. Fermentation is doing what? Where you have an organic molecule, right, as both your donor and your acceptor for electrons, right? But here we have an inorganic molecule that's actually our receptor, which is oxygen. So respiration generates significantly more energy than fermentation. This actually was important in evolution. Why? Because of that rise in energy, organisms could become more complex. They have the ability to make more energy so they can do more things, right? And so complexity kind of come about with the same rise in aerobic respiration. So why oxygen? It definitely is available, right? So as it increased in availability and became really abundant, organisms just decided to take advantage. They were like, okay, that's great. Also, power, highly electronegative. So here's kind of a look at the TCA cycle metabolism, right? Like I said, 2 carbons come in. We'll talk about all the steps. Just so you know, sulfur now is a lot less commonly available than it was back in the beginning, right? So oxygen, powerful oxidizing agent, has a high affinity for electrons, large energy release with each electron that is transferred from a carbon to an oxygen. So that's an interesting thing, right? Is every carbon that gives an electron to an oxygen, there's this big burst of energy, this big release. So, oxygen, how do we start getting there? Cyanobacteria. So what are cyanobacteria? They're photosynthetic bacteria, okay? So photosynthetic bacteria, we talked about photosynthesis just real briefly. What does it do? It is the formation really of sugars, right? But as a byproduct, it gives off oxygen. So it takes in carbon dioxide, makes some sugars, gives off oxygen, and cyanobacteria started releasing this oxygen out about 2.3, I think now, usually in microbiology, I say about 3 million years ago. But it didn't accumulate in the atmosphere for several hundred million years. But modern organisms, that's how we classify them now, is based on their ability to both handle and detoxify oxygen species. So let's talk about these. Why? Because this could be a good matching, could be a good fill in the blank, okay? So, what are we? We are obligate aerobes. So obligate aerobes, I'm going to drop to the bottom. Why? Because we can't suddenly stop breathing, okay? We have to take in oxygen. So obligate aerobes, dependent on oxygen for energy production. We do have reactive oxygen species protection, right? One of the proteins that I work on, I'm always talking about it, superoxide dismutase 1. At the end of you making ATP, right, we have an oxygen radical produced. And superoxide dismutase 1 comes in and breaks that down into hydroperoxide, which is also bad for the cell. And then catalase comes by and breaks that down into just water and oxygen. Okay? But we do have detoxifying molecules. We also have GSH, which I think people may have heard of or not. It's also a detoxifier. But we have many enzymes and kind of antioxidant molecules. Obligate anaerobes. Obligate anaerobes are just the opposite. They cannot have oxygen there. So if you take microbiology and you're digging in the soil for your sample, right? We always tell them go dig out in the soil for sample. I love teaching microbiology labs. It can be very fun. It's nice to see what people come up with. But if they dig really far and they plate it, sometimes they get nothing, even though we know there's tons of bacteria. Why? They dug so far, they ended up with a bunch of anaerobic bacteria. Sometimes they can be kind of in the middle and you can get facultative anaerobes where they can kind of deal with oxygen, but they don't really want to. Okay, so they just don't grow as well. So obligate anaerobes, no oxygen. They do fermentation. So remember, fermentation again is organic molecules for both donors and acceptors. Aerotolerant anaerobes, they still do fermentation, but they have the ability to detoxify it. Oxygen facultative anaerobes, they have the ability to detoxify and can use oxygen if it's around, right? So the soil got stirred up or something, you can have some facultative anaerobes that can kind of deal with it and you utilize it. So those are the different classifications based on oxygen usage and detoxification. So let's talk about aerobic metabolism. What is that going to be? TCA cycle. ETC, your book breaks it up where the ETC and oxidative phosphorylation are separate. Back back in my day, we just learned it as complexes 1 through 5, and they're still complexes 1 through 5, right? So just one thing. Because it goes through those things on the 4 complexes and then goes to ATP synthase. We certainly wouldn't want to stop at 4. Occurs where in the mitochondria? So we were already starting to move into the mitochondria last time when we said pyruvate's brought into the mitochondria, right? So occurs within the mitochondria. Free energy is captured by the electron transport chain. Why? Because electrons are then transferred from redox pairs with more negative reduction potential to those with more positive reduction potential. Other than just knowing that, you don't have to know anything else about reduction potential. I'll give an example later. I'm going to put it in your help. Okay? So, redox coenzymes. I love to talk about coenzymes. I think they're pretty cool. What are coenzymes? We said all coenzymes are cofactors, but not all cofactors are coenzymes, right? Because cofactors could be metals and other things that are not organic. But coenzymes are organic molecules. Okay? And so it would also be considered a what? Cofactor, but it is a coenzyme. Now, redox coenzymes for aerobic metabolism, we've already talked about NAD, because we saw NAD before, and glycolysis. So, what we have nicotinamide adenine dinucleotide, and NADP, right, with the phosphate group on there. Okay? Both structures have adenosine and an N-ribosal derivative, so sure, and a nicotinamide linked together. They call this through a pyrophosphate group. You can see kind of the 2 phosphates right there for each pyrophosphate. See pyrophosphate group. The difference between the NADP is there's a phosphate like hanging right here. Okay. Now, riboflavin. This is one of the things that they check for when you come into the doctor's office. They check your B vitamins. Right? If you don't have really good B vitamins, what happens is you're tired. Okay? So B vitamins are very important. Why? Because they're important a lot of times for the formation of all these molecules. So riboflavin B2 is a component of 2 coenzymes, both FMN and FAD. Okay? So FMN, what is FMN? Instead of FAD, where you have dinucleotide, here you have FMN, which is mononucleotide, flavin mononucleotide. Flavin comes from riboflavin, right? I always put a look at the name. Mono, obviously. Okay? So flavin adenine dinucleotide would be FAD. You're going to see these in the electron transport chain. So in glycolysis, we've got NAD only, really, and NADP. Here you're going to see the FADs. Why? It takes less energy to move things over to FADs. Right? Remember I said that NADs were a little bit higher energy molecule. It takes more to reduce those and do things with them than it does for FAD. So here we're just trying to save a little energy. So we use FMN and FAD. Now, they can be bound as prosthetic groups, so that's when they're bound, to flavoproteins. Okay, so I would know the term flavoproteins. They can act as both donors and acceptors of 2 hydrogen atoms. Now, the interesting thing about the mono is it can transfer 1 hydrogen at a time. Now, this is important because it links up the TCA cycle to the electron transport chain later. The TCA cycle, we had 2 pyruvates, right? So we had 2 acetyl-CoAs come in. So we actually have 2 things moving at a time, and we're moving multiple electrons at a time, even on top of that. And the electron transport chain, you're actually only moving 1 at a time. So we needed something that can kind of make that transition. So this molecule is one of those that can accept from the TCA cycle, but then just move it 1 at a time to the electron transport chain. Okay? So these have roles in both TCA and ETC. Citric acid cycle. All right, series of reactions to release chemical energy stored in the 2-carbon. Like I said, 2 carbons coming in, 2-carbon acetyl group is acetyl-CoA. Acetyl-CoA is coming from pyruvate. It also can come from fatty acid catabolism, because remember we said fatty acid catabolism does not give you what? Sugars. All right? So we can't make sugars from fatty acid breakdown, but we can come into the TCA cycle to make some energy. Okay? So fatty acid catabolism, you can also do like some amino amino acid breakdown as well, right? Sometimes we have excess amino acids, we kind of need to send stuff through the TCA cycle, we can do that as well. Composed of 8 reactions, and we have 2 stages again. Stage 1 is going to be the first 5 reactions, like what we saw last time. And stage 2 is going to be reactions 6, 7, and 8. And 6, 7, and 8 are only to regenerate the stuff for reaction 1. Okay? So it's just to regenerate the oxaloacetate. So you can see 2 carbon acetyl groups of acetyl-CoA are actually going to react with that oxaloacetate made from step 8. And then, like I said, in the second phase, oxaloacetate is regenerated. So, first reaction that's going to come in is we have an introduction of 2 carbons as acetyl-CoA. Oxaloacetate, what is it doing? It's reacting with acetyl-CoA and water. So we end up with citrate. Citrate and CoA. Now, this is an aldol condensation followed by hydrolysis. We're using water to break something, right? Who is catalyzing this is citrate synthase. Synthase uses ATP? No ATP. No ATP. We're going to have one later on that's called a synthase also that actually does use ATP because I'm going to tell you all about that one. It has 5 names. So it just depends on what your book wants to use, right? So this is citrate synthase. Okay? Now, oxaloacetate first condenses with acetyl-CoA to form citral-CoA, which is what you're getting right there, and then it's hydrolyzed and we got water. Water coming in, hydrolyzed to citrate and CoA. Okay? Now, citrate, you can see that first reaction is very what? Exothermic. Thank you. So, if something's highly exothermic, what do we think is going to happen with that reaction? It's irreversible, that's good. What else? Regulated. So highly regulated. So this is going to be one of those regulatory steps. Okay? So highly regulated and going to be irreversible. This enzyme, what is it actually doing? It's removing a proton from the methyl group of acetyl-CoA and it makes it an enol. Remember we said enol formation is real commonplace in some of our pathways because we want something that is what? Unstable, right? We want something that has a lot of high energy in that molecule so that it'll help pull things forward. So the fact that it forms an enol, that enol attacks the carbonyl carbon of oxaloacetate, and that's kind of what helps push this thing forward. Okay? That's just me getting into the mechanisms a little bit more. Now, what do we have after that? We have 2 carbons coming in, so that's what you're going to know. 2 carbons coming in, and we form citrate. 2 carbons come in, citrate. Okay, what do we have after that? We have citrate to isocitrate. Okay, citrate to isocitrate. How do we do that? A conatase. We are now newly working on a conatase in my lab. Let me tell you, it is a go-to activate. Why? Because it is an iron-sulfur cluster protein, and iron is very reactive to oxygen. And so it has been very difficult to keep it working. But the enzyme conatase, more formally known as a conjugate hydratase, most people just call it a conjugate though, catalyzes reversible. So you can see here the arrows are going both ways, right? So you can actually just go back to the beginning and come back out. But reversible transformation of citrate to isocitrate. How this happens is through kind of an intermediate form of tricarboxylic acid cisaconate, and that's just through a dehydration. So basically, you're taking the water off and then you're putting the water in these 2 reactions that you're seeing. So, carbon-carbon double bond of cisaconate is rehydrated to the more reactive secondary alcohol isocitrate. So what are we doing? We're making it more unstable, more reactive. Alright? Why do we do that? Again, just to make things more energetically favorable for the second step, but is this reversible? Yes, it is. It looks like it's very bad, right? It's like plus 13.3, but we saw this in glycolysis test tubes, right? Again, we'll get measured at points plus 13.3, but physiologically it's a lot closer to equilibrium, and it's kind of pulled forward by the other reactions that are surrounding it that are favorable. Okay? So, when we have 2 carbons come in, formation of citrate. What do we have after that? Citrate to isocitrate. Okay? Then what do we have after that? We have citrate to alpha-ketoglutarate. Okay, so oxidation to alpha ketoglutarate. I think we called it last semester oxidation to alpha K. Okay, oxidation to alpha K. So oxidation of isocitrate to alpha ketoglutarate. What is this going to give you is not only is going to give you an oxidation. We said oxidation what? In order to take something off, you got to put it somewhere else, right? So this is going to be one of our energetically favorable steps because we're making NADH. Okay? So NAD or NADP, doesn't matter whichever one. It gives us NADH, reduced NADs of some form, and carbon dioxide. Isocitrate dehydrogenase. We've talked about dehydrogenases before. Those are oxidizing enzymes. What do? They dehydrogen, right? Dehydrogen. Okay? So, catalyzes oxidation and decarboxylation. We said decarboxylations were also energetically favorable. We saw that before. Isocitrate to form alpha ketoglutarate. The oxidative decarboxylation actually is going to occur. It's going to be stepwise. Do you need to know necessarily all the steps? Probably not, but I would just know that isocitrates oxidize to oxalosuccinate. The decarboxylation is actually what's driving a lot of this. The decarboxylation of oxalosuccinate to an enol intermediate. So a lot of these have enol intermediates. Okay? Why? To give rise to the reaction, right? Just to help make it energetically favorable. Now that enol is going to rearrange to an alpha K. That rearrangement and that decarboxylation are both going to give rise to a good delta G. Okay, now this is prime, so this is inside. Okay, so delta G non-prime of like minus 21. Okay, so that's favorable. So reaction 1 was favorable, reaction 3 was favorable. Now, as it is favorable, guess what? This is going to be what? A step for regulation. Exactly right. So this is going to be a regulatory step also. Okay. Now, in the glycolytic pathway, we only have one oxidation. Here we have quite a few. So we have another oxidation right behind this one. So we came in. 2 carbons came in. We had citrate. We went citrate to isocitrate. We went isocitrate, oxidized to alpha ketoglutarate. We're going to have alpha ketoglutarate. It's going to get oxidized to succinyl-CoA. Okay? Succinyl-CoA. Now here, because we have another oxidation, what are we going to do? We're going to reduce NADs. Okay? So now we have another reduction of a high energy molecule. So we're making lots and lots of energy. Okay? Oxidation of alpha K to succinyl-CoA. You can see that this is highly favorable. As being highly favorable, guess what this is? Regulatory. So this is a regulation site again. Yeah.
How important is it to be in the regulatory since it's like both ways we go back and forth, but we're making power application, right?
Yes, we are. This also, we're going to talk about this pathway. TCA cycle is interesting because it's not just catabolic. It actually is very highly anabolic. It feeds into other things. It makes things. So all of these intermediate steps are actually precursors to your amino acids. So it could stop right there and just put this one out and make something else. You can say, it could say, okay, well at this point in time, we have this succinyl-CoA, but I need to make all of the amino acids that are based off of succinyl-CoA. Alright, so that's a good question. So we're going to talk about this again, kind of like a hub for both anabolism and aging pathways. Okay. So oxidation of alpha K to succinyl-CoA, got another NADH produced, another carbon dioxide form. So this is another decarboxylation on top of that. So highly, highly favorable again. Alpha K is converted to succinyl-CoA. Who is doing this is alpha ketoglutarate dehydrogenase complex. So this is actually a complex. It's 3 enzymes working kind of together. Okay? Would I know that? Yes. Why? Because alpha K, it's a cool complex. Okay? What does it have in it? It really has 2 dehydrogenases and then something to move something around, a transsuccinylase. Okay? So one of those is actually the alpha ketoglutarate dehydrogenase, right? So one of them is the alpha K dehydrogenase. One of them is dihydrolipoyl transsuccinylase, and then a dihydrolipoyl dehydrogenase. So 2 oxidizers and one that's just going to move things around. Okay? So I will just know the 3 components of the alpha K dehydrogenase complex. So what do we have? We had 2 carbons come in, right? Y'all be with me? 2 carbons come in. Can y'all say it? 2 carbons come in. 2 carbons come in, and do we form what? Citrate. Then we do citrate to what? Isocitrate. Then we do isocitrate to what? Oxidation to alpha K, right? Then what do we do? Oxidation to succinyl-CoA. 2 carbons come in, citrate. You can say citrate synthase, right? 2 carbons come in, citrate synthase, right? What do we have after that? Citrate to isocitrate. What do we have after that? Oxidation to alpha K. What do we have after that? Oxidation to succinyl-CoA. So we got a lot of oxidations in this one. Okay, this isn't even all of them. We're going to have more oxidations in just a little while. Okay? Reaction 5, conversion of succinyl-CoA to succinate. Succinate's a big time molecule. It goes off lots of places. Okay, so it has lots of potentials. Succinyl-CoA to succinate, we cleave it. So this is actually a cleavage. We cut it. So kind of like we have a cleavage event, right, at step 4. In our glycolytic pathway, we have a cleavage here. Cleavage of succinyl-CoA is coupled with what? Our first SLP. Okay? So now, we have SLP, right? We're paying ourselves some money, and we get what? Cleavage. Okay? So cleavage, SLP. Cleavage of what? A high energy thioester bond, where we have it right here. You see that sulfur group right there? Right, on succinyl-CoA. Cleavage of that high energy thioester bond forms succinate. It is reversible, even though it's high energy. It is catalyzed by what? Succinyl-CoA synthetase. I don't call it that, I actually call it succinate biokinase. So that's why I put that in there. What that means is, but in mammals is interesting because we have 2 forms of this. So we talked about ATP and GTP, and I said, are they energetically equivalent? Yes or no? Yes. Right? I said, it didn't matter what the phosphates were stacked to as long as the phosphates were there. This is actually how we make GTP. We have 2 versions of this succinate, or this succinyl-CoA synthetase. We have a GTP version that will SLP GDP, and we have one for ATP. So mammals have 2 versions, okay? One that will put the phosphate group on GDP, and one that will put the phosphate group on ADP, okay? So keep that straight, because mammals are special, okay? So in mammals, coupled to substrate level phosphorylation of ADP or GDP, we have 2 enzyme forms expressed in most of our tissues, okay? One for ATP, one for GDP. Now, if you look at this one, it actually goes by lots of names, not even just those 2. Some people even call it succinyl-CoA synthase. Okay, because it depends on which reaction you're going for, since it's fully reversible, whether or not you're using ATP or not, whether you're doing some kind of hydrolysis or not. Okay, so just know that's the reason behind it. If you're reading a book or you're looking at something on Google or what have you, you might see it as other names, but it's still this guy. Okay, it's just depending on which reaction you're talking about. And so that is reactions 1 through 5.
Dr. Boyd,
Uh-huh.
I have a question on reaction 5.
Uh-huh.
Even though it has like a negative delta G, but it's like comparatively like lower compared to like other regulators, this one's higher regulation?
This one's a little bit less.
So like the more negative you go, the more high.
Definitely. This one, it's kind of more of a regulation like product-wise, right? So whoever's around like afterwards, as opposed to like having other allosteric sites and things like that for this one. But yes, good question. But we'll even talk about regulation in just a minute, because I'm going to give you all those little slides that talk about who's regulating who where, right? So this is the completion of the TCA cycle. We're doing oxidation of succinate to oxaloacetate. And it's basically 2 oxidations with a hydration in the middle. Okay? Now, I want you to know these 3 reactions. Why? Because if you're taking Biochem 2, you'll see them again. If you're taking microbial physiology, you're going to see them again. Okay? These 3 reactions are very important because you see them also in breakdown and synthesis and amino acid breakdown and synthesis. And y'all get into amino acid biosynthesis and biochem 2 or microbial physiology. So you'll see these three reactions again. What are they? They're an oxidation from a single bond to make a double bond. We took a hydrogen off, right? We made a double bond. Once we made that double bond, what do? We actually added water across the double bond. So it's a transhydration where we added a group sticking out this way, and we added a group sticking out this way. Okay? And then what do? We oxidize again. Back to a single bond. Okay? So an oxidation resulting from an alcohol to a ketone. Okay? So what is it? Oxidation, transhydration, oxidation. Oxidation, double bond, transhydration across that double bond, oxidation again. Okay? So what do we have coming in? We had 2 carbons coming in, and then what do? We formed what? Citrate. Citrate, synthase, you can say whatever, right? What was after that? Citrate to what? Isocitrate to what? Oxidation to alpha-K. What was after that? Oxidation to succinyl-CoA. What was after that? Cleavage, SLE. So we ended up with just succinate. Okay? What did we cut off? CoA. Alright? So we got succinate. Now, what are we doing with that succinate? So we're right here, right? So we cleaved this thing off, right? And we said, okay, now we're going to end up with succinate. So let's talk about what's happening. Succinate dehydrogenase. So this is that first oxidation to make a double bond. Succinate dehydrogenase is an FAD-dependent oxidation. Remember we had biotin-related enzymes before, we had to have biotin there, right? Here we have something that is bound to an FAD. You have an FAD bound in the active site. So FAD-dependent oxidation of a single bond and succinate to a double bond to make fumarate. Mechanism involves hydride removal by FAD and a deprotonation. This is not exergonic enough to reduce NAD. Remember I said NAD is a higher energy molecule, so we need FAD in the electron transport pathway. This is one of those that talks to the electron transport pathway. This enzyme is actually embedded in the membrane. This is in the intermitochondrial membrane, or in the mitochondrial membrane. Why? Because it is part of the electron transport chain. So it's embedded in the intermitochondrial membrane. But it also has a part that sticks out into like the interface, right? So it talks to the cytosol as well. And so it's part of the TCA cycle as well. So it's kind of bridging the 2 pathways. The electrons transferred from succinate to FAD to form FADH2 are then passed on to ubiquinone and the electron transport pathway. We'll talk about ubiquinone more as we get in there. FAD is covalently bound to who? Succinate dehydrogenase. How is it covalently bound? A histidine. Okay? Bound to a histidine. The covalent bond between FAD and succinate dehydrogenase is going to link, you don't have to remember which carbon, the C8 carbon, or C8A carbon to FAD. Now, what do I want you to know? We have an FAD bound to a histidine. That's what I know. Okay, right? We had a phosphohistidine intermediate. Some of our previous reactions here, we have an FAD bound to histidine. Histidines are really important in active sites. We've already seen that before. What is FAD? FAD looks a little bit different than NAD. So here we have a flavin derivative, riboflavin B2. It is required for synthesis of both FMN and FAD. They are found as tightly bound prosthetic groups. When something's really tightly bound, covalently bound, then it is a prosthetic group also. So not only is it a coenzyme, but it's also a prosthetic group. I know we're throwing all kinds of things out there, right? And it's also a cofactor, right? I say those things so that you don't get confused in case the wording as you're studying is one or the other, right? They're all interchangeable in this case. Now, required for synthesis of FMN and FAD, these are found as, again, tightly bound prosthetic groups, and flavoenzymes. If you have an FAD or FMN bound, you're considered a flavoprotein. Flavoprotein. What are flavoproteins? They're basically just redox proteins. Okay, they're just doing reductions and oxidations. They're just redox proteins. Okay. Why do they use flavin coenzymes. Why? Because they can actually be in different oxidation states, so this becomes useful. So they can exist in 3 different oxidation states. And this allows flavin coenzymes to participate in both 1 electron, which we're talking about for FMN, or 2 electron transfers at a time. Why is this important? Because we said that complex 6, right, or the reaction 6, right, that succinate dehydrogenase reaction, right? We said the interesting thing about it is that it's embedded in the membrane for the electron transport chain. I said the TCA cycle, you're moving 2 electrons at a time. The electron transport chain, you're moving 1 electron at a time. So you need to be able to have something that can talk to both pathways. So these molecules become very important because they have the ability to move them 1 at a time. Okay, so we can participate in both 1 electron or 2 electron transfer reactions, and because of that, flavoproteins are really responsible for a lot of biological systems reactions. Work with a lot of donors and acceptors, and they're basically your main classification of redox enzymes. Okay, flavoproteins. Now, kind of backtracking a little. That's just because we just talked about the things that are stuck to succinate dehydrogenase, right? But I mentioned aconitase. So I kind of want to go back. What was aconitase? Step 2, really, right? So aconitase, I told you that it's been a booger bear to work with in the lab. Why? Because it has iron sulfur clusters. Iron, we talked about iron, what happens to it whenever it gets oxidized, right? It no longer binds oxygen, right? Because of that hemoglobin, right? Whenever you have it down there and it's gone to iron 3+, right? Here, the interesting thing is that as I'm working with it, if it gets oxidized, I can't get this protein to be activated. So, we have iron sulfur clusters, and succinate dehydrogenase not only has an FAD, right? Not only has a flavin derivative bound to it, a prosthetic group, it also has iron sulfur clusters inside of it as well. Okay, so we'll be talking about it in respect to succinate dehydrogenase and also aconitase. Succinate dehydrogenase, it has all 3 types. It has a 4 iron 4 sulfur center, a 3 iron 4 sulfur center that makes kind of a little cube that's also seen in aconitase, and a 2 iron 2 sulfur center. These clusters, they only cycle back and forth between 1 state. So what are they good for? Moving 1 electron at a time. Okay? So iron sulfur clusters, we use them in our electron transport chain just to move 1 thing at a time. Okay? So typically cycle between iron 2 and iron 3, right? 2+, 3+. So ferrous and ferric. SDH is bound, succinate dehydrogenase, is bound in the inner mitochondrial membrane. The electrons from the FADH2, they enter the ETC via the iron sulfur clusters, and they will actually contribute to ATP synthesis at the end. Okay? So this pathway, the TCA cycle, once you go to step 6, it actually starts feeding into the electron transport chain already. Okay? It still continues to do that oxidation, dehydration, oxidation, but it also is feeding those electrons from that oxidation into the electron transport chain. Waste not, want not, right? So if you're going to do all these oxidations, you might as well send them through. Okay? So that is something about iron sulfur clusters. You're going to see them a lot. Here's kind of a look at some iron sulfur clusters. What would I want you to know? These distances between iron sulfur clusters, man, that has really evolved over time to be like peak performance. So they are very optimally spaced to have the correct distance between them so that we have really good transfer. So distance of redox centers in the protein is optimized to facilitate electron transfer. Multiple redox centers, like what you see here, kind of all in a row, we call those an electron wire. Y'all, I like to ask that. Why? Because electron wires are cool. You're going to see several. Okay? So electron wires are kind of cool. It just kind of bounces along that pathway. Okay? What else do we have? Back to reaction 2, aconitase, because I didn't want to talk about iron sulfur clusters just yet. But here we have citrate to isocitrate, right? We said citrate to isocitrate. has a secondary alcohol, which then can be oxidized. So if you're looking at it, what happens is alkanitase removes pro-RH of the pro-R arm of citrate, and how it's doing this is just by using its iron sulfur cluster, because it has a strong affinity for those things, just to move those things around. What would I want you to know? Alkanitase has an iron sulfur cluster. I don't even think that we get that deep into it. Yeah, we get. It's a 4 iron 4 sulfur cluster. Where I'm getting stuck is I have 3 irons there and all my sulfurs there, but I can't get that last iron to snap in. So until the last iron snaps in and forms the whole cube, it's inactive. My iron keeps going +3 before I can get it snapped in. Okay? So this contains a 4 iron 4 sulfur cluster. It's bound by the side chains, like I said, 4 iron 4 sulfurs. Who's got sulfurs? Cysteines, right? The thioneins also have sulfurs, and you'll see them later. But it's bound to side chains of cysteines, okay? Now, binding of iron to the vacant position of that cluster is what activates it. So like I said, I can actually purify it with 3 of the irons already there. It's getting that last iron to kind of slide down in to complete the little box. That's kind of a bugger, okay? But putting that last one in that vacant position is what actually activates it. The added iron then coordinates the carboxyl and hydroxyl groups of citrate and acts as a Lewis acid. So that's how it's able to do all of this to make secondary alcohol, accepting an electron pair from the hydroxyl group just to make it a better leaving group. Do you have to memorize all of that? No, just know that we have a 4 iron 4 sulfur cluster and a conitase. That citrate's not really good about being oxidized, and so we have to use this method. Now, you can jump back around. So, what do we do? 2 carbons came in, and we form what? Citrate. Okay, 2 carbons come in, citrate formation. Okay, 2 carbons come in, citrate formation. What's next? Citrate to what? Isocitrate. What's next? Isocitrate to alpha K, right? So oxidation of citrate to alpha K, right? Now, what do we have after that? Oxidation of alpha K to succinyl-CoA. What's after that? Cleavage and SLP, right? Cleavage and SLP. What's the interesting things about that? We have 2 versions of that enzyme, right? One will stick the phosphate groups on ADP, one will stick the phosphate groups on GDP. Okay? Now we have 2 versions. Okay? Now, what's after that? Oxidation of succinate, right? So oxidation of succinate, we said we end up with a double bond. Yes? And that double bond makes us fumarate. So oxidation of succinate, fumarate. Okay? Fumarate. It's interesting because we have a fumarase. Fumarase catalyzes transhydration. So you can see you're basically just adding water kind of across the bond, double bond, okay? Transhydration, a fumarate to form what we call L-malate. We even kind of briefly mentioned malate before when we talked about trying to pull things back out of the mitochondria so we can do anabolic reactions, right? So, malate. Hydration occurs right across that double bond. Transition of water. The actual mechanism for this, as of yet, is not fully elucidated. Okay? We're just not real sure. Several hypotheses out there. They may have gotten it narrowed down a little bit more now, but just no, not really sure. Okay, last step, malate dehydrogenase. So this is that last oxidation. I said we had an oxidation, transhydration, oxidation. This is that last oxidation. Okay, what's this oxidation? We're going to oxidize malate. Malate to oxaloacetate. This is an NAD-dependent oxidation. Carbon that gets oxidized is the one that actually got the secondary alcohol put on it just a minute ago. Okay? And we did that transhydration. We're just oxidizing that OH group. The reaction, look at it. Is it good? No. It's actually very costly to make oxaloacetate. Very. And the previous reactions were interesting because they were kind of helping happen close to equilibrium. So they're not helping draw drive this. So what is actually pulling this reaction forward is step 1. Step 1 is paying for step 8. Okay, because it's cycle, right? So step 1 pays for step 8. So energetically expensive, because delta G naught prime, that's in the body, right? That's a physiological pH and everything else. We're looking at plus 30. That's bad. Okay? Now, the concentration then in the mitochondrial matrix is actually pretty low, right? Pretty low. Yeah, that actually helps push it forward too, because concentrations, right, will help bring things forward. The other reactions the way it needs to make it, because the product amount is low. Yes?
Is it also very costly, that gluconeogenesis part, where we convert from malate to cell, that's why they go back?
Yeah, anytime you're converting to oxaloacetate, it's expensive. But again, it's kind of driven by those next reactions to go back to okay. But yeah, that's why I stay always expensive. However, malate dehydrogenase reaction pulls forward by citrates and things, right? So reaction 1. It was plus minus 33, right? So it helps pull out. Plus the fact that the product is at a low concentration all of the time, right? We said that low product, what it's doing, it helps shift that equilibrium, right, where you want to pull through. And so keeping that product concentration kind of low also helps assist in pulling this reaction forward. So oxidation of glucose is going to be a 24 electron oxidation. So electrons from glucose oxidation kind of feed into the ATC pathway. What does that do? Gives you the ability to make ATP. More than 30 ATP are made per glucose molecule, depending upon which shuttle you use to get it out. It could be 32, it could be 34, okay? On how many ATP you get back out of one glucose molecule. And that is per glucose molecule. That's pretty good, right? Because when you're busy sucking on that Jolly Rancher, that's not one glucose molecule, that's a lot, right? So you're busy breaking that down and making at least 30-something ATP per each one of those glucose molecules. So that's the TCA cycle. TCA cycle, 2 carbons came in, 2 carbons come in, right? Start there. 2 carbons come in. 2 carbons come in. Say 2 carbons come in. 2 carbons. What next? Citrates in this, right? Or citrate. Okay, 2 carbons come in, citrate. Alright, what's after that? Citrate to isocitrate. Last groups were like citrate to isocitrate, you cis-aconate. It don't matter. But citrate to isocitrate. Now what's next? Oxidation to alpha K. What's next? Oxidation to succinyl-CoA. What's next? Cleavage and some pay, right? Cleavage and some payout. Okay, what we do is just cut CoA off. Okay, so cleavage and some paymates. Interesting thing, should be, and you have 2 enzymes for that, right? And we got cleavage and some payment. What do we end up with? Succinate. So now what do we do? Oxidize beta double bond with fumarate. Then what do we do? Transhydration. Then what do we do? Oxidize again, now we're back to oxaloacetate. So oxidation, transhydration, oxidation. Yes?
Did we use any ATP for the cycle? No, we just made one. Even the costly last one, that wasn't without ATP for making cell acetate.
For oxaloacetate formation, no. Why? Before it will with the first reaction. That's who's paying for it. The synthase reaction. Alright. So yeah, you're just getting stuff out this time, which is nice. Alright, pyruvate. We always said 2 carbons came in, but we didn't really talk about the 2 carbons coming in. We just said we had 2 carbons coming in, right? So pyruvate, how did it get in there? Okay? We actually decarboxylate it. So pyruvate is decarboxylated to acetyl-CoA. Acetyl-CoA then comes into the TCA cycle. Remember we said decarboxylation is energetically good, right? It's nice. So decarboxylated to enter, okay? For each glucose, you have 2 acetyl-CoA. Remember, because you have 2 pyruvates, right, for each glucose molecule. Now, in eukaryotes, that would be you. Pyruvate goes into the mitochondria. So pyruvate goes into the mitochondria, and then all of this takes place. Where does this take place? Mitochondrial matrix. Write that down. Mitochondrial matrix. What is it? Basically the cytosol of the mitochondria. Okay? The mitochondrial cytosol. So the mitochondrial matrix, yes. It is. Yeah. Yeah, it's decarboxylated and then comes in. So, after acetyl-CoA comes in, right? No, it's brought in. Pyruvate, yeah, it's brought in that way. But yes, that's what we brought in. Yes.
Before it goes to my
Yeah. So pyruvate actually can enter mitochondria.
matrix or just intermembrane space?
It can enter the intermembrane space.
But not that.
That's where the matrix is. Intermembrane space is what got in it.
Is it that
It's
intermembrane
solution, right? But you're talking about matrix and the crystate, yeah, it's the same thing. So you can do things in the intermembrane space, but you do have stuff there. Yeah. And that's going to be buried in the intermitochondrial membrane. That's in the intermitochondrial membrane. All right. We're going to see the whole thing put together with exactly how this comes in. Okay? So, pyruvate. We said pyruvate is transferred into the TCA cycle. TCA cycle happens in the mitochondrial matrix. Now, we have 2 ways to get pyruvate in there. So there's actually 2 pathways for this. And we're going to talk about both pathways in just a minute. Okay? So, pyruvate transported into the mitochondria. All the downstream reactions occur there. We have 2 different positions for this. We'll talk about pyruvate dehydrogenase, and we'll talk actually about a different carboxylase that brings those in as well. Okay? Fatty acids can come in at the level of acetyl-CoA. So I said fatty acid breakdown, metabolism can actually come in as well. It'll come in at the form of acetyl-CoA. Each cycle, what do you need to know? Each cycle will yield 2 carbon dioxides, okay, 2 COs. It'll give you 3 NADH. Why? Because you had 3 oxidations, right? And this is per molecule, okay? Per molecule, there's 2 of them, so you're going to have 6, right? Per molecule. 2 carbon dioxides, 3 NADH, 1 FADH, 2 and 1 ATP, right? That's whenever you did the substrate level phosphorus. Okay? Y'all good? All right. 2 carbons come in. Citrate synthase. Citrate to isocitrate. Isocitrate, we're going to do oxidation alpha K. Oxidation of alpha K to succinyl-CoA. Succinyl-CoA, what are we going to do to it? Cleave the CoA off and pay ourselves a little ATP. Okay? Or GCP, whatever it is, right? Then what are we going to do? Oxidize succinate, right, to fumarate. So oxidize, double bond. What are we going to do after that? Transhydrate, right? Transhydrate. Then what are we going to do after that? Oxidize again, right? And then we end up with oxaloacetate. And we said oxaloacetate kept in very small concentrations to help pull everything forward and is really predominantly paid for by reaction 1. Okay? After that, let's talk about what you get, and we're going to talk about pyruvate dehydrogenase. All right, glycolysis and TCA cycle net reaction. This is what you would look at. If we go through both things, right? From glycolysis, you swallowed at a jelly rancher, each molecule broke down. You have 10 NADH, 2 FADH2, and additional 34 molecules of ATP in both the electron transport and oxidative phosphorylation processes, and you add glycolysis all the way through. Okay? Now, TCA cycle. What's it important for? And the reason we're going to talk about how pyruvate gets in here is because pyruvate is our central metabolic hub. It can come in, it can go to something else, right? But it comes in different ways. So we're going to talk about both of those in just a minute, as well as the things that come off that are generated from the TCA cycle intermediates. Now, TCA cycle, we consider this the hub. Okay, this is the hub of both anabolic and catabolic processes in your body. Pretty much every intermediate that is made feeds into some kind of feeder pathway for some amino acid biosynthesis, fatty acid biosynthesis, and biosynthesis, okay? So pretty much every one of these goes somewhere else. So you can see here that we have everything coming in. Pyruvate, look, pyruvate itself can actually be giving rise to valine, leucine, alanine, all kinds of other things, right? It can drop down to acetyl-CoA, and then we said even fatty acids can come in at the point of acetyl-CoA. Steroids, right? Acetyl-CoA can be used for steroid formation. Now, if you look down, citrate also has places to go. Isocitrate does too, but alpha-ketoglutarate is one of the big ones. So as you go into Biochem 2, they actually call it the alpha-K family. Okay, the alpha-K family is all of your purine nucleotides and glutamine, glutamate, ornithine, citrulline, and arginine, with proline kind of off to the side. Proline is one of the derivatives of it. Okay. Then you go down to succinyl-CoA. You get all of your succinyl-CoA family, which is glycine, gets fed into there actually to form with succinyl-CoA and make 2-amino, 2-ketoadipate, and then also aminoebulate. It gives us 4 rings. So see, like I said, all of these things are biosynthetic for amino acids or some kind of biomolecule. Why do we need 4 rings for? Heme, right? You like the 4 rings, right? They hold kind of things in place. Everything, right? So we have to have 4 rings for those things, and so that gives rise to those. Then we said, okay, let's look. Look at those last 3 steps, right? I said we're making oxaloacetate, but even in those last 3 steps, we have a lot of feeders. Fumarate and oxaloacetate actually go into the asparaginous aspartate family. Okay? Do y'all have to memorize all of these? No. What I'm trying to tell you is that this pathway is really responsible for not only energy formation, but also the formation of like pretty much everything that you need to survive. Okay, so the TCA cycle, very, very important. So you can see asparagine, aspartate, phosphate, aspartyl semialdehyde gives rise to threonine, isoleucine, lysine, methionine at the end, right? So very important families. Now, here's the bad thing. How do we make sure that we're not using it all up? So how do we know as we're feeding through, right, whether or not we need to send it all the way through to generate the full amount of energy, or whether or not we need to feed it off into making all of those amino acids, because you're constantly making proteins, right? So we're making proteins, we're repairing our DNA, we're doing all kinds of other things in which we have to have anabolic reactions. So how do we make sure we don't use up everything in the TCA cycle? How we do that is by doing what we call anaplerotic reactions. Okay? Anaplerotic. What does anaplerotic mean? It just means we're filling them back out. These kind of prevent you using everything up to where you can't go through the rest of the TCA cycle. Yeah, you have to have oxaloacetate. Even in small concentrations, we have to have it. Okay, so we have to eventually send something all the way through. Okay, we can't just drop off at all these other positions and not continue through the cycle. Okay, so anaplerotic reactions. CO2 provides the actual carbon source for the anaplerotic reaction. So far we've been using a lot of organic molecules, but here CO2 is going to do it. Pyruvate carboxylase, which we talked about, as part of our 1 and 1.5 reactions going back for gluconeogenesis. That pyruvate carboxylase is also part of this. Amino acid metabolism, so breakdown of amino acids, actually is used to fill back up this pathway as well. Right? Let's say you decide you don't need that much methionine or lysine or whatever, you can feed back in and go back to fumarate. Okay, to make sure that you're going back the other direction and can fill up and go to oxaloacetate. So all of these can feed back in. So amino acid metabolism can also provide back to the TCA cycle intermediates. For animals, that's you. Pyruvate carboxylase, we talked about pyruvate carboxylase, PEP, carboxylase and carboxykinase. Pyruvate carboxylase is actually your most important anaplerotic reaction. Why? Because it's part of glycolytic pathway in the TCA cycle, so it can be in. Okay? Now, pyruvate carboxylase, it is activated. So now we're talking about activators. acetyl-CoA. Okay, so it's activated by acetyl-CoA. When acetyl-CoA exceeds the supply, so this is a feed-forward mechanism, right? So when acetyl-CoA exceeds the supply of oxaloacetate, so when we have way more 2 carbons coming in than what we have after our A reaction, right? We have way more than that. Pyruvate carboxylase is stimulated to help kind of make more oxaloacetate. So the interesting thing is we don't have to go through steps 6 through 8. We can actually backtrack and feed directly into the oxaloacetate. Okay? So how do we do that? So if you look up here, we need to look up here.